So thank you all for coming and the volunteers for making this happen, and for the people under 20 to spend a Wednesday evening to hear about HIV. I know it's not the most cheerful topic. I'm really grateful that you came. I'm going to talk fast because I've got way too much to present to you tonight. And I'm an evolutionary biologist, a virologist, and I am an immunologist. And those three things come together to try to help us design a vaccine. So my talk is going to start with Darwin and some history, because Darwin is where my work begins, actually. His 200th birthday was a few years ago, so there was lots of talks all over the world to celebrate it. And I got to give some of them, and my friend painted this for me. But there's the man. At any rate, we're going to learn a little bit about what he did first. So Darwin just had an incredible insight, and it wasn't born out of nothing. His father actually was a botanist and had done some plant breeding, but they didn't really think about what it was that they were doing. And so he went on a long sea journey, and he went to the Galapagos Islands, and you guys probably have heard this story, but he noticed that finches were very distinctive, and the way they were distinctive partly was by having really distinctive beaks that made them able to survive better in the microenvironments they were in these islands. And so he got the notion that they might have evolved to have changed to really adapt to their settings. And this fundamental notion is really the fundamental aspect of biological science today. I think we all use it. And I love this particular thing. This is a notebook of his, and this is a very famous phylogenetic tree. And he wrote, "I think," and then he drew the first phylogenetic tree. And this is my bread and butter. I do phylogenetic trees all the time. So this was just scribbled in his notes. "I think," and then this shows relationship between things. And you guys might have seen or used family trees before. They're kind of similar. We'll talk about that in a second. But what people had before they really understood DNA and how DNA related to all of this was they had phenotypic characteristics. And so they would try to group animals by phenotypic characteristics, and animals that shared, or plants that shared particular aspects, you would group closer together, and you'd think that they were more related. And so you could draw this kind of a phylogeny, and you'd think that they have diverged over time to have these different attributes. So you can see like mammals and dinosaurs would be different parts of a phylogenetic tree. And this is the way you read them. You have groupings that are closer together, like that little clade A there is a group that's kind of close together. Clade B is another group that's close together. And it goes further back in time before they diverged. And you have a root where everything beyond that root is shared. And you often will fix this whole thing and outgroup something that's distant from everything else in the tree. And then you have something called a most recent common ancestor. And this is something actually that we make vaccines out of, because it's very central to everything that emerges in the clade after it. So with viruses, when we make one of these trees, we actually can use these trees for many practical purposes. Retroviruses, which HIV is one of, actually doesn't have DNA as its genetic material. So all of you guys have DNA as your genetic material. It has RNA as its genetic material. And what these viruses and viruses do in general is actually very insidious and kind of creepy. We're made up of cells, right? So a virus will infect some kind of our cell, and it will integrate its genetic material into ours. So actually, with a retrovirus, you start with the virus getting into a cell, and then it's got RNA in it. That RNA is copied to DNA. The DNA goes into the nucleus and then is inserted right into our own genomes. And that DNA encodes information that will turn the cell into a virus-producing factory at that point. So it's no longer a healthy, good cell useful to us. It's a cell that's carrying this virus and producing more and more virus. So what winds up happening is that the RNA gets copied from the DNA, and that makes protein. You guys might be familiar with that very fundamental chain of life. And then the proteins assemble and pick up a whole new viral RNA genome. It gets packaged, and it buds off a cell. So this is a picture of actual HIV viruses budding off a cell, carrying their genomic material out. So when I draw phylogenetic trees, they're usually over a very short time period. maybe a course of one person's infection, or maybe the course of an epidemic in a country. And this is an example of how HIV varies, and it's really incredible. So this is an alignment of DNA sequences, and every time you see a little tick, it's a mutation in the DNA. And all these red and green guys here are from a single donor. So all of these guys are from one donor, and this little branch represents how many ticks or how many mutations there are from everything in this branch. So these guys are all identical or nearly identical. This guy might be, 10% apart or something. So this is like what everybody gets after they've been infected for, wondering if I've got, no, I don't have a pointer. After they've been infected for some years, your virus evolves within you. So every single person who carries HIV doesn't carry just one HIV. They carry a swarm of very, very diverse HIVs. And then when they transmit to their lover or their partner, one of those comes through. And this is a newly infected person who is infected by this person. And their virus right at the beginning is extremely homogeneous. It doesn't have hardly any variation. And then the immune system hits it and it starts to vary. So this happens in every single one of the 33 million people who are infected. And so every time it passes, you get some random new virus. So everyone infected is carrying a different virus. And those mutations are immunologically relevant. They're often escape mutations from the immune system, so they matter to the immune system. So we have to make a vaccine that's going to work against all of these global variants. That's the challenge, and is why we don't have one after all these years of effort, but there's some reasons for hope. So one of the things that we have up at Los Alamos is a database that's really serves the world. So every HIV scientist can go to our database and pull out all the available sequences, lots of information about them, everything that we can get from the immune response. So these are phylogenies. Those are phylogenetic trees too. They're drawn a little bit differently. So the root is in the center, and all of these are different sequences isolated from different individuals. We have 50,000 subjects represented there that we've gathered from all of the literature from all over the world. Then we spit that back out to the world. And I was supposed to give one of these lectures last year, but our database was down, and we have millions of users, and I felt terrible about it. So I didn't give the lecture because I was trying to get our database back online. We'd been hacked, actually, and so the security guys took us down. So we had about 2 months we were down, and it was 12-hour days for the team. But anyway, this is something we do for the world. And this is the basis of my research. So I use all of the sequences gathered from all over the world to try to make a vaccine that will work against these diverse viruses. So back to Darwin just for a minute, because I think this is so interesting. In 1964, Howard Temin got the Nobel Prize for discovering retroviruses. And he was actually one of the guys who founded our database. He thought we needed such a thing because retroviruses were so variable. They thought we needed one place to keep track of them. But actually, as it turns out, we haven't just descended from monkeys, and this was a parody about Darwin when his last book was published that came out in Punch's Almanac. But anyway, so they were mocking him for thinking we descended from monkeys. Actually, we also descended from retroviruses. They're a significant part of our genome. So about 10% of our DNA comes from retroviruses. We've had a long, long history with these beasts. So we and our ancestors have had to live through and get through different retroviruses through the years. And because they integrate into our DNA, we can continue to carry them. And so it's kind of an interesting way to think about descent, but there's all sorts of funny things about evolution that you might not think of initially. And this is just one example of a retrovirus impacting our own DNA, but there's many, many such examples. So this IRMG gene was in rodents, so distant ancestor of a distant relation of ours, and it got turned off. And so old and ancient primates have this gene turned off, but there's a retrovirus that came in and inserted and reawakened it. So more recent common ancestors with us have this gene awake, and in fact, this gene is an important part of our immune response. So these things aren't just silent, and they're not just buried, they're actually part of us and how we work. So what can we add to our understanding of history of this epidemic? through modeling viral evolution. You guys who are in your teens here have lived with this epidemic your whole life, but some of the older folks in the audience know that we didn't have to worry about HIV until 1981, when it was first described in Los Angeles. So guys started getting cancer. They thought it was a very unusual kind of cancer for young men to be getting. They realized something was causing this. Their immune systems had been really severely damaged. They didn't know what was going on. And a couple years later, they isolated the virus, 1983. And as soon as they had the virus isolated that caused this disease, they were able to produce diagnostics to tell people if they had it, and they were able to start educating people about how not to transmit it and how to have safe sex and how to use different needles if you're going to be a drug user. So they started educating people about prevention. 1986, a few years after that, we'd had now a few viral sequences, and my mentor at the laboratory, a guy named Gerald Myers, realized that this virus was incredibly diverse, not like anything that he had seen before. So he went to the National Institute of Health in Washington and he said, we need a special place to keep all of these diverse genes, and we need to be able to track them and help other people study them. And so that's how we got our database funded. So that happened in 1986. It wasn't until 1997, so 16 years later, that we had the first effective treatment. And again, some of the folks in this audience might remember having a friend or a loved one or a family member who got HIV. There is nothing you could do. It's a lethal disease and you can't be cured of it. You can have it suppressed by drugs, and that's what we do now, and it's wonderful. But you have it for life. And if you don't get drugs, you'll die of AIDS in about 8 to 10 years. So it was a long time that we had to wait before we got effective therapy. And by 2009, we had 38% variation in envelope, 33 million people living with HIV. So the phylogeny and the evolution of the virus can help us fill in what we don't know from earlier, from the time it was discovered. Why didn't we know that this disease existed until 1981? Well, we know that as soon as it was discovered, it was already very, very diverse in Africa. So it had been in people for a while. And then in the mid-90s, my colleague David Ho in his laboratory went back and looked at old frozen samples in Africa, and they found one sample from 1959 that actually was positive for HIV. So we know that one gentleman living in the Democratic Republic of Congo had HIV at that time point. And we also learned that many primate species in Africa carry SIVs, which are simian immunodeficiency viruses related to HIV. So every monkey has its own kind. And these SIVs are common in bushmeat. So if you go to the market in parts of Africa where monkeys are eaten, you'll find bushmeat, and the bushmeat will have SIV in it. So there are changes in Africa. Colonial practices really could have exacerbated its spread. There was needle reuse, so they were giving people like blood infusions and other ways of using needles, and they just would use the same needle over and over again, and blood is a good way to transmit HIV. Urbanizations, so people were coming into the cities. Forced relocation, wars, deforestation, maybe more exposure to primates through the process. So these things all changed, and so this thing that had been with the monkeys in Africa for hundreds of thousands of years had opportunities to come into the human population in the last century that they'd never had before. So all of these different kinds of African monkeys carry HIV. It was the chimpanzee that had the form that established the epidemic, the global epidemic that we face now. But there's also been gorilla SIVs that have moved into man as well. And you can see this all through these evolutionary trees and the relationships in the DNA. You can trace this. So this is just to point out you don't necessarily, there's a lot of discussion of sex with monkeys and all sorts of things back in the 80s and 90s. You can just imagine that a cut finger or something like that could easily lead to transmission. So this is now one of those phylogenetic trees that shows relationships in DNA of many, many different kinds of SIVs. There's something called HIV2, which is common in West Africa, and came from sooty mangabe monkeys. And then there's HIV1. So all the diversity in HIV1 is represented by those little red symbols. And the chimpanzee and the gorilla viruses are all up here. So we've got the tip of the iceberg from this very diverse, very ancient viral expansion in Africa among primates. Here's our little 1959 sequence, and it's closer to the center of the tree, so it's more like older sequences than any of these modern ones. So we knew it was old. So now we can start filling this question mark in. We not only know that there's a first positive sample, but we're able to take all the contemporary samples and try to work backwards and figure out when was this virus first introduced, or when was that most recent common ancestor that began to expand in man? When you look at that, it turns out that it was around 1930, maybe 2 decades or so before that. And some of the clades that we have, like the B clade is in the United States and the C clade in Southern Africa, probably had their origins about 1916, really began to expand. You can see that when you start modeling the rates of evolution, there's probably a very, very slow expansion first. Probably it came into somebody, started getting transmitted in rural areas, maybe not transmitted very fast. Maybe it wasn't very good at getting transmitted and adapted. And then it began to really expand at a high rate. So other people have looked at this same question. That was data from my laboratory that I just showed you before. But a group, Variability and Walensky, used different techniques, and they came up with a slightly pushed back estimate of the time depending on how they looked at it, or if when they looked at it kind of like we did, actually quite the same time frame. So it's been with us for probably about 100 years. Doesn't mean that it hasn't transferred from chimps more times than that, but the one that caused the expansion in humans, that most recent common ancestor, the Eve, if you will, of the HIV epidemic, was about 100 years ago. So we know that it started slow, gained speed. We know about when it started. And there was another sample that was found from looking at archived tissue samples from 1960 that was also found in the Democratic Republic of Congo. So now this is the Democratic Republic of Congo sampled in 1997. That's the diversity, where each of these branch links again represents a lot of changes in the DNA. This is one city very early in the epidemic. This is a single individual 6 years post-infection. So you remember that trial, that genetic diversity I showed you? This is one person. This is many, many people after about 70 years. And this is what happens when you have a new introduction in a community. This is a global sampling of an influenza epidemic in 1996. So we've got influenza vaccines that work quite well, but this is the diversity that they have to cover. You have to change it every few years because one of these grows out and repopulates the next flu season. But this is about what a vaccine has to deal with. And you can see that it's about comparable to the HIV diversity in a single individual. So it's a really challenging problem. When I think about a vaccine, and when my colleagues do, we try to think about a global vaccine. And this is the diversity in Africa. Remember, I was telling you that this region of Africa is probably where it started from. So these are clades. These are these genetic groupings of viruses. So South Africa and Southern Africa is almost all one clade, so it's less diverse. But Central Africa, probably where the origins of the epidemic are, and it was older, is very, very diverse. And I really want to try to make a vaccine that could handle that. That's what we're going for. HIV doesn't only evolve by point mutation, but also by recombination. So if a person gets infected with 2 of these different forms, they can recombine within that person and make a very different virus. So it has lots of tricky ways to evolve. I'm showing this machine just because I thought it was so beautiful, but it is a 10,000 sequence tree, and it's got a lot of different individuals in it. And you can kind of tool through this tree if you have the computer running, and each color set's a different person. But anyway, one of the things that was nice at Los Alamos is that we did have supercomputing facilities that were free to us to use, and we were able to build these phylogenies, which are pretty complex models for many, many sequences, and that's been quite useful to us in the past. So what we're up against, HIV's got a generation time of 2 days, a reproductive ratio of about 6, so if one virus is going to produce 6 more live ones, an error rate, so every time it copies itself, it makes a mistake. So it has this great pool of diverse viruses. to be selected from. We've got a generation time of about 28 years, so we're a lot slower. We've got a very precise copying mechanism of our DNA, so we don't evolve so fast. But we have this beautifully clever immune system that can see this world of diversity when it's in us. And we have really polymorphic genes. Everybody carries different immune genes in their bodies. All of us do. So we see viruses in different ways. And we use combinations of these genes to create the immune response. And antibodies even evolve during the course of a response. You make an antibody response to a virus or a pathogen, and that antibody keeps evolving. It doesn't just sit still. So it's evolving while the virus is evolving. So how does the immune system work? I'm going to now show you a highly technical video. If you don't remember anything else from this talk, you should get this. This is it. Oh, shoot, I can't get it. Let's see, we were making it work before. So let's see. I don't know why it was working before and now it's not. Ah, there it is. It was working. It is going, but it's not showing. He's got me in a different system here. It was just, I'm gonna end the show and try to start it again. Maybe I can't. Okay. Nope. Let me try one more thing. Oh, shoot. So it was definitely working before.
Let it run once.
Oh, okay. It was working just fine before. Yeah, it was fine.
Maybe you could open the wrong file and make some information.
I don't. He had it. It didn't look like this. I don't know. He's got this thing where I'm seeing the slides ahead that he didn't have originally. Huh. Well, I'm disappointed because I wanted to show you. I've got two videos for you that I wanted to show you.
Do you not have the raw file on your hard drive?
I do. Yeah, that's an idea. Okay. I'll have to find it, though. I'm sorry, guys. I don't know. He's changed the display just before he left. Okay, here it is. Let's see if this works.
If you hit the space bar, it should come up big.
Okay, so this is an example of how the immune system works. You have something foreign coming in, something that shouldn't be in the system. And you have something that can recognize that it's not you. And it's armed. So it's able to take care of and kill invading organisms. So you guys are probably doing this as we speak in your bodies. And I'm just going to go here and see if we can move on. Okay, so I'm going to try to show you a little bit how this works. And you can see why I wanted to show you the video. Okay, so the first thing that happens when something foreign comes into your body is you have an innate immune response that recognizes foreign. And so you get molecules that recognize this and bind to things that are different. And they cause the first cells, which are things like macrophages. Macrophages means a big eater. It binds to foreign things and it engulfs them. And then when it does that, it eliminates them, but it also chops them up and presents them to the rest of the immune system. So a toll signaling pathway is part of this, and I just put this in because I think it's so interesting. Das ist ja toll was a German scientist who discovered this gene, and without it, flies don't work quite right. But they also don't have a good immune system. They can't see fungus, and so this is a fungally infected fly. And we have a related gene to this in a very, very ancient part of our immune system that can see foreign right away as soon as it comes in. And I've got another movie, which I also want to show you. I'm so sorry, I just got to. So I'm going to stop here because I think this movie is very cool. Also, maybe.
That one looked like it played, yeah, try.
Oh. No, it's not playing. What this is it's macrophages in action. And so, this is also going on while we speak in your bodies. And it's really pretty incredible. They pick up, shoot. Now I don't even know where I am.
One behind the finder is where you found the files before.
Yeah, I can't get to that from here. It's not on my screen anymore. Well,
Two displays are different.
yeah. And I don't know how I got into this one. Well, he gave me something different than he left me with, than I practiced with.
Try moving the cursor off to the right.
Do you want to try?
Okay,
Have a go for it.
let's see.
Okay, so let's go there because we can get the movie.
Okay,
So you want to go to phagocytosis movie. There we go. So let's stay there and play it again for me a couple times, because it's very impressive. So you guys all know how to do this. You're doing it all the time. So these are macrophages engulfing yeast. So if you got something like yeast infection, they would say, okay, I know this isn't me. Go ahead and play it again, if you can. Thank you so much. So there, I just got one. And they have these wonderful little like pseudopods that reach out and feel their whole environment looking for things that are foreign. And then when they find one and their receptors are woken up, they engulf it. So that's the first step, actually, one of the very first steps in the immune system. So let's go back to the PowerPoint now. My wonderful audiovisual aid here. Bless your heart. So it'd be PowerPoint Café Scientifique.
Does that work?
Yeah, I think that works. Okay, all right. Okay, so you've got these professional antigen-presenting cells, and they also destroy some of these early invaders. And so these are these cells that can engulf something, and they not only engulf it, but they chop it up inside, and they start summoning, they start secreting chemicals that summon other macrophages and other immune cells to a point of infection. So this is just a picture of all of the macrophages getting summoned to clean up a viral infection on the liver. So this is what happens. But this isn't all that happens, because these macrophages, after they've chopped up the invader, display bits of it on their surface and wake up something called a helper T cell. And this is really the conductor of the immune system. This guy, when he's woken up, calls all of the cells of the immune system, stimulates antibodies, stimulates other kinds of T cells, and says, we've got trouble. We've got to stop this attack of a foreign invader. So what happens with the macrophages, they carry bits of the foreign proteins in something called a class 1 molecule. And we've all got these molecules, and they're very, very polymorphic, so we have different collections of them in our bodies. And we carry different little fragments out, and they get carried out to the cell surface. And T cells will recognize something foreign in the context of this class 1 molecule. And when that happens, you have a naive T cell, and it's been woken up, and it's seen something different, and it's very specific for that little tiny fragment. And it starts making an immune response. So it starts dumping out chemicals that summon the rest of the immune response, and some of these cells go into memory. And when they go into memory, they wait till you see that infection again. So if you've been infected with something like Zika virus, you're safe for the rest of your life. You won't be infected with Zika virus again because you have these memory cells. So if Zika comes back into you, your body will recognize it and attack it really fast and clear the infection. Antibodies are a little bit different. They don't see these little fragments of something foreign. An antibody sees a whole protein. So this is the envelope protein of HIV, that red and gray thing up here, and this is an antibody. And the antibody surface recognizes the protein surface of the HIV. And the thing that we're seeking in a vaccine is something called a neutralizing antibody. So here's an HIV envelope coming in ready to attack this poor T cell because it wants to turn this T cell into a viral production plant. But this antibody is going to get in the way, and it binds to it very specifically, and it won't let HIV in. So these are the kinds of antibodies that we're trying to make, but we have to make an antibody that's able to see this world of HIV diversity. So how does a vaccine work? Well, what happens? If you're a vaccinologist, my partners, who are the experimentalists who work with me, will take a killed virus, maybe, an attenuated virus, so it's been made weak so it can't make you sick, or a non-toxic small part of a virus, or a pathogen, a bacteria, and they give you that as a vaccine. So they get something that looks just like the terrible pathogen, but it doesn't get you sick. Your immune response can see that thing, and it can remember it. It can remember it for years, decades, maybe your whole life. So it's a very natural thing. Your immune response isn't going to wear out because you've got too many vaccines, because you're seeing all sorts of foreign things all the time, and you're keeping safe from them. Your immune response is very powerful. And once you've been vaccinated, you're primed and ready if you ever encounter the live pathogen. So what happens with that immune memory is as soon as you encounter the live pathogen, you can make a very active and aggressive response. You don't have to wait for the whole immune system to come up again. So that's the goal, and vaccines have been a huge triumph for mankind. I think of, getting to the moon is a beautiful thing. I think of getting rid of smallpox as a great and powerful thing. Smallpox is a disease that killed 300 to 5 million people in the last century. Very ancient disease. It's been with us since we've been. Those who did survive, many died. When they got it, they were often severely scarred and often blinded for life. And the very last case of smallpox in our world was October 26, 1977, because of vaccines. Edward Jenner was the person to think of vaccines. And what he noticed is that milkmaids didn't get smallpox. Everybody else was getting smallpox, but not milkmaids. And there was a cow named Blossom in 1796 who had cowpox. And Blossom had a milkmaid, Sarah Nelms, and she got cowpox. And when you get cowpox, you get a little pock. You didn't get what you just saw on the other slides. So Sarah visited Dr. Jenner with her mild case of cowpox. And Dr. Jenner had this notion that the cowpox was going to keep her safe. And so he got a little pus from the pustule on her hand. And he caught the gardener's kid, James Philp. Don't know, you could not do this these days. So he took young James and he scratched his arms and he rubbed pus into his arm, carrying the cowpox. And then he tried really hard to give James smallpox. He kept exposing him over and over. And he had, I don't know how they did this, but how the gardener put up with it. He's paying him to do the vegetables. But anyway, he was right. So James was safe. He never got smallpox. And so this was really the first successful vaccine. And just to give you guys a sense, I love this paper. It's an old paper, but it shows you how much good they've done in our country. These are all old vaccines that we've had before 1980, and these are how many people got the disease, how many people died of the disease. After we've had the vaccine for a while, how many people got the disease and how many people died. It's remarkable. And I have a very hard time understanding why people don't like vaccines at this point in time. They've done tremendous good for us. This is vaccines that are not as old. And at the time this article was written, they still hadn't been able to clear out the diseases, but they were already doing a great deal of good. You can see number of cases has dropped hugely, and number of deaths has dropped hugely. And I don't know where these vaccines are at now, but this is the right trend. So just to give you some recent data with influenza, what they do is they take people who are vaccinated and people who aren't vaccinated, and they see how well they do. They use some measure of performance. And flu vaccine reduced children's risk of having to go to intensive care by 74%. So kids who got the vaccine were much less likely to get very sick. And elderly people are really fragile when it comes to influenza. People 50 years and older who got a flu vaccine had their risk of getting hospitalized decreased by 57%. So people don't get as sick when they have the flu vaccine. And the people who are at the highest risk are little kids and old people, and they do much better if they get the vaccine. But they're not the only people who are getting the vaccine. You know, people in the mid-range are too, and that vaccine can protect people around you. So my husband and I had a kid with very, very weak lungs, and our doctor made sure we got the vaccine every year so we wouldn't bring it home to him. So it's got value for the people around you as well as yourself. It's a community thing. Okay, so HIV, the way it gets into our bodies is through these T cells, these helper T cells that are so useful to the immune system, and it eventually takes them out. So after 10 years, you don't have your helper T cells anymore, and you're susceptible to all kinds of things. Could someone give me the time? Thank you. Okay. So these beautiful T cells that are the conductors of our immune system, the virus gets in them, and here's a T cell that's been infected. These are all viruses coming out of it. So it's no longer doing its good T cell work, it's making viruses. So you want to be able to kill those T cells. And we have a lot of good news on the AIDS front. Antiretrovirals, the drugs you give to people who have HIV, don't clear the virus, but they bring down the viral loads and they keep people healthy longer. And they reduce HIV transmission to their partners. So if you've got HIV and you're taking antiretrovirals, you can live many more years with health, your immune system isn't decimated, and you're also much less likely to give the virus to your partner. So there's many good reasons for taking antiretrovirals, and now that WHO and CDC and everyone recommends that everyone should get them from very early on. As a result of this, AIDS deaths are down 2.2 million in 2005 to 1.2 million in 2015. So we're on the right track just with the drugs. But why do we still need a vaccine? Well, there are 34 million people living with HIV, and it's a lifetime infection. They're never healed. So if they stop the drugs, the virus just comes right back very, very quickly. So you have to give 3 to 4 drugs every single day for life. If you go off the drugs for a while, the virus comes back and you start getting drug resistance. Universal access to therapy, this is expensive therapy, is really challenging. So currently, less than 50% of those who need it are getting it, but it's getting better every year. We're getting more and more. Part of the problem is that this disease is most devastating in some of the poorest countries, and these are very expensive drugs. But in our country, we still have 1 million people living with HIV, and a lot of people living with HIV don't even realize they've got it because it's a chronic infection. It takes a while to kill you. So you might be positive, infecting other people, and not even know you have it. It's really challenging because it's so variable, and the adaptive immune response is really specific. So remember, I was telling you, T cells see specifically a pathogen, and then they remember it. Well, if you've got different pathogens in every person, you've got to make an immune response that's able to see all of them. So HIV is changing and evolving all the time in every single person, by base mutations and recombination, which we talked about. It also does by insertions and deletions, and it modifies its glycosylation sites. So very often my computer screen looks like this. My husband thinks I'm crazy. For 20 years I've been staring at these things. And these are all mutations in proteins. So clones can be variably expressed too. So you might have, in HIV, you might have a single clone, and that clone, when it gets in a cell and it gets to the surface or it gets put onto a virion, might look different. It might have its sugars added differently. It might change its conformation a little bit. So there's not only the genetic variability, but there's some epigenetic variability. So even the clones can be different. So I want to make a multi-clade vaccine, and something that I came up with in 2007 that my colleagues and I worked together on to try to realize, this was actually a very difficult project and took a village, was we got all of the sequences together that we could in the world, and we started recombining them and evolving them in a computer. And we wanted to solve the problem of if we could only have a couple proteins to put into a vaccine, because every time you put another protein into a vaccine, it costs millions of dollars, and your funders don't want to do it. So the fewer number of proteins you can put in, the less expensive the product is going to be. But we wanted to, how do you pick the proteins you put in then? Well, the proteins I picked, we evolved in a computer to be the proteins that could give the best coverage of HIV diversity in the world. So rather than just picking 4 natural proteins, we made 4 proteins that looked and felt like real HIV proteins, or 3 or 2. You could set the number that your funder is going to allow you to make. And you say, okay, for these 2, if I can only have 2 proteins, these 2 proteins give you the best coverage of HIV diversity possible. So that was the idea, and I'm not going to go over this because I'm running out of time, but this actually works pretty well. So we came up with the idea, had our first paper in 2007 with theory, and then a couple of labs tested it. This is the Baruch lab at Harvard's test, and these, we call them mosaic proteins. So this is the number of T-cell responses to heterologous different strains that we're able to get with 2 mosaics, and we're able to get, each one of these is a different monkey, and each monkey is making a bunch of responses, a bunch of good immune responses. If I used the ancestral state of HIV, that most common ancestor, so I could reconstruct that from a tree, it doesn't exist anymore. And in fact, this is an approximation of it. But if I make an approximation that's central to all HIVs, I'm able to get more cross-reactive responses than I am with a single natural protein, which is what people usually use in a vaccine. So these are all different monkeys with a single natural protein. And these are 2 natural proteins. So we're able to get a lot more responses. So that was good, because we knew that in monkeys, if monkeys can be infected with SIV, so that's the animal model that we use. We take a macaque and we can give it SIV. They'll develop AIDS. And so we can see we can protect monkeys with our vaccine. And we knew that if you made a lot of responses, the black guys are the guys that were actually protected, the monkeys didn't get sick. So the immune response was strong enough not to block the infection, but to contain the infection, and the monkey stayed healthy. So the guys who made the monkeys that made lots of immune responses are these guys, and they didn't die. The monkeys that were red didn't get a vaccine, and this is how quickly they died. This is days following infection. They died quickly. A vaccine that didn't work quite as well, didn't give as many immune responses. They lived a little bit longer, but the ones who made a lot of responses lived a long time. So we thought that making a lot of responses is a good thing. So then now we tested it with a challenge. Again, my colleague Dan Baruch at Harvard did these tests. So now he takes a bunch of monkeys, and sham means they were not vaccinated with the HIV. They were vaccinated with just an empty vaccine. And the other 2 both got the mosaic that we designed at Los Alamos. And here you have rates of infection. So what they do is they take a monkey, they vaccinate it, and then they try to infect it. And they hit it a bunch of times. And you can see the monkeys that didn't get the vaccine, the black ones got infected really quickly. So most of them were infected the very first time they were challenged. But the monkeys that got the mosaic vaccine could be challenged many times before they finally got infected. And that's great, because when you have sex with somebody who's HIV positive, it usually takes 100 sex acts before you pass the virus. It isn't every single time. And so if you can really slow down that rate of transmission, that would be great. You'd have many fewer people infected. But not only that, once those monkeys finally were infected, because they kept hitting them over and over again, they lived. So here are the monkeys that were infected that didn't have the vaccine dying. Here are the monkeys that got the vaccine. So it really was able to inhibit infection and slow it down, and it was also able to give a good enough immune response so it lived. So we're excited about this vaccine, and it's in human trials right now. We're getting the very first results out of a mosaic vaccine that's being conducted in the United States. And they're just looking for immune responses and safety right now. So they've given it to about 120 folks. They're all done. But they have to do the analysis now to see what their immune responses were like. These are not people at risk for getting HIV. They're volunteers. Usually somebody who's lost someone they love to HIV will volunteer for this kind of thing because they really care. So you give them the vaccine and you make sure it's safe, and you see what their immune response is like. And what we're looking for is an immune response like what we got in the monkeys, because it does very well in the monkeys. So if the humans do that, we don't know that it will, doesn't always, but if it does do that, then they'll move this into a huge efficacy trial. But it'll depend on the results. And I'm getting emails about this weekly, and it's looking good. The mosaics seem to be giving more intense responses. We've got mosaics in several other trials as well that are less far along. There's also pretty cool new strategies for T-cell vaccines. One is called a CMV vector. So a vector is something that carries a vaccine in. You put the HIV into the vector, carry it in. And the CMV vector is really changing the way we think about immunology, totally changing it. Our paradigms are shifting, seismically shifting, because it's using all kinds of new immune responses we didn't know about 2 years ago. So there's been a lot to learn through this vector. But this vector is the only thing that's ever cleared SIV. So what happens in the monkeys that get a vaccine carried in this vector is that you give them a vaccine, you do that infection thing where you keep hitting them. When they get infected, you can show they've gotten infected, but their T cells click in and they clear the infection 50% of the time. No one has ever cleared an HIV infection, and no monkey's ever cleared an SIV infection. So 50% healed monkeys is quite remarkable. And so people are very excited about this. The vector they need to make safer before they can put it in a human trial, but Lionel has designed the HIV part of that vaccine, and they're hoping they can maybe treat people with it. They're hoping, we don't know yet, but that maybe you can give an HIV vaccine to someone who's already infected in one of these amazing vectors that's stimulating all these novel kinds of immune responses, and maybe you can heal them. Yeah?
CMV cytomegalovirus?
Yes, it is.
Is that the one that 90% of school-age children have by 5?
We probably all have cytomegalovirus in our history. So what happens, though, in very rare cases with pregnant women, it can cause a microcephaly that's sort of like what Zika can cause. So they want to make it a virus that cannot do that. They want to make it super safe for everybody. So we all have it, and it doesn't bother most of us most of the time. Very rare cases that can happen. One thing you might do is say give it to men. It's sort of a hard, or give it to women post-pregnancy age. So those kinds of things might be a possibility. But they're just trying to make it as safe as they can and be sure that that doesn't happen, and maybe they can give it to everybody. So that's what they're working on right now, is safety issues.
I read somewhere, this was years ago when I was still in school, that cytomegalovirus can help the HIV virus actually cross the blood-brain barrier. Is that true?
I don't think that's true. I don't remember such a thing. You've got a beautiful memory if you're remembering that from years ago. Good for you. So yeah, I think there are issues with cytomegalovirus, but I also think that if they can attenuate it adequately, that it's really got some chances. And if you had HIV, you might want the vaccine, even if you were, because if you could clear the virus 50% of the time if you got the vaccine. I mean, this is all conjecture because it's only been done in monkeys now. A guy named Louis Picker figured this out, and our kind, human beings, have been living with cytomegalovirus forever, and it's been living with us. It's an enduring infection, also persistent. So it's learned how to handle the immune system. So even by studying this vaccine, we're learning so much about the immune system the cytomegalovirus is teaching us. Yeah. Yeah. So people are also considering antibodies as prevention. And you guys, I'm going over time. Do you want me to stop, or you want me to go on?
Go.
Cool. You got it. Thank you. Okay, so people are also considering antibodies as a prevention strategy. So instead of trying to make a vaccine stimulate antibodies, you take good antibodies from somebody who's been infected, you clone them, you make a bunch of that antibody, and you maybe put that in a gel or something for prevention during sex, or you might give those really good antibodies to a person who's infected and see if that could help clear the virus from that person. So there's a lot of interest in that right now. We're working very hard on that with some of our partners. If you did that, remember I told you T cells have memory? So they go into this latent thing and they just sit there waiting until the virus that they can recognize comes and that wakes them up again. These same T cells can carry HIV. So that's why when you give drugs it doesn't clear. There's these latently infected cells just lurking that are carrying the virus that are hidden from the drug. And you stop the drug and then they wake up sometimes. As soon as they wake up, they repopulate your infection. So you'd probably want to do this with something called a latency reversing agent, which wakes up all of these latently infected cells, and you hit it really hard at the same time with very effective antibodies. So that's one goal. The number 3, it seems like 3 would be a good number. And this is just because these are different antibodies, and these are 200 different viruses up the column. So every little band is a different virus. And these are really good antibodies. And if they're bright red, they can really kill the virus well. And if they're white, they can't kill the virus at all. So this antibody kills these guys really well, these guys not so well. And these viruses, it doesn't touch at all. This antibody hits these guys really hard. So if you hit with 2 antibodies, both of them keep working, and you can cover all of these viruses with at least one of them. If you added a 3rd one into the mix, you can really get incredible coverage of many, many diverse viral forms. So that's part of why we think we've got to have 3. With Ebola, it turns out that the only thing that's been effective so far at treating Ebola is 3 antibodies given like this. And it's not very effective in humans. It's kind of effective in monkeys, and a little bit effective in humans probably. So it required 3, though, and it was very hard to predict the optimal combination. So that's another thing we're working on in LANL, is how to predict optimal combinations of antibodies for treatment. And I'm just going to talk very quickly about Ebola. We're also working on Ebola vaccines. Ebola and Marburg are related viruses. They're in the family called phylogeny. And they've just been with us, that we've known about them since 1967. So a monkey shipped to Germany caused an Ebola case in Germany, and the person died horribly, and we recognized we had a new disease. The first Ebola outbreak was in 1976, and since then we've had about 50 of these outbreaks, mostly in Africa. It's a very ancient virus family, and it's not mutating quickly like HIV. So what happens is you get an outbreak, and the outbreak is distinctive, kills a bunch of people, and then dies out. But in 2014, we really woke up because we had this terrible outbreak that killed many, many people in West Africa. So there were close to 30,000 cases and 11,000 deaths in that outbreak, and we were all aware of that from the news. So people are paying a lot of attention to Ebola now. And we don't even know very well what the animal reservoir is. So this virus is in an animal reservoir, and every once in a while it comes into humans, and we have one of these outbreaks. We know that bats carry this, and we think that bats are responsible for some of the outbreaks. And we know that primates also can be infected by Ebola. In fact, really tragically, Ebola is really damaging the gorilla communities, for example, which are endangered and beautiful animals, and they have a lot of trouble with this virus too. These viruses are transmitted through body fluids and needles or something, and that's it. They're not airborne or mosquito-borne, but they can be readily transmitted through body fluids. So these are the outbreaks we have sequences from, and this is another one of those phylogenetic tree, and this is where the different outbreaks have occurred and the year they occurred. But what you can see when you look at this tree is EBOV has called, it's the Ebola virus. EBOV stands for Ebola virus, but these are closely related. They've caused outbreaks as well. These guys are all really similar. So if you make a vaccine against an EBOV, it's going to work against the other EBOVs. And that's probably true. Problem is that every once in a while you get, like, this is Bundibugyo. And Bundibugyo's caused 2 outbreaks. They've been lethal and horrible, 2012 and 2007. So you don't know what the next one's going to be. You don't know if it's going to be EBOV. You don't know if it's going to be a completely new one we've never seen before, or one of these rarer ones. So again, the same kind of philosophy, what I want to try to do is make a vaccine that would react against this whole world of filoviruses and protect against any one of them, since we don't know what the next outbreak will be. And so my husband and I have just been working on such a design, and this is the proteome of the filoviruses up at the top, so all the proteins stuck together. And what we did is we picked out the very most conserved parts of the proteome. So those are the red bits. So those parts are present pretty well conserved in all of the filoviruses. So these are our targets, and we glue them together and we make a fake protein, and we make a couple of mosaic-like proteins. We've got a kind of a second generation called epigraph now, but it's a similar theory, different mathematics. And what winds up happening, this is the vaccine that they're using now, and it's an Ebola vaccine, and this is the coverage that it would give you of other Ebola viruses, but it would not cover Marburg viruses. It wouldn't even cover very well other Ebola virus family or genus viruses. And if we use our vaccine design, this is the kind of coverage we can get. So if we make an immune response to our vaccine, it would react with all of those viruses. And that's what we were looking for. So this thing, where we are with this right now is we just got the theory paper accepted. So it's in press, and we've given, about a year and a half ago, we gave the design to our colleagues at Oxford at the Jenner Vaccine Institute, and they vaccinated mice with this, and it's working well. So the next step is to talk someone into funding a monkey study and a challenge study. Yes?
If you did put different types of herpes, like maybe CMV virus in an ape or a monkey, and it activated those new immune responses that you're discovering, what would that do to this model? Would it like make better antibodies?
Well, this is actually a T-cell vaccine.
Oh, oops.
And in fact, the CMV one is a T-cell vaccine. So the one that's so remarkable, that's kind of changing our whole way of thinking about immunity, is particularly about T-cells. Most effective vaccines in the history of vaccinology, though, have been antibody. So it would be kind of different if we go into this zone. But again, we've never seen anything besides the CMV that can clear HIV, or can clear SIV. We don't know yet. It can clear HIV. Yeah.
Sorry, you might have said this already, but was the Jenner Institute named after that person who worked with
Yes, it was. Yes, it was. That's right. He was English, and the Jenner Institute is at Oxford, and it's their vaccine institute. Yeah. Thank you for pointing that out. Okay, so that's the end. These are just a handful of the wonderful people I work with at Los Alamos. James Seiler's my husband, and he's a physicist and a mathematician, and I have to convert everything to X and Y's. He doesn't allow any of this T-cell epitope stuff, but if I do that, he does beautiful work with us. And Simon Perkins, a while back, worked with James, and he actually wrote the Mosaic code for us. Will Fisher, Karini Isam, Peter Haber, and Chidit Jwa were all postdocs with me, who are all now staff scientists at Lannel, and they're a great group of people to work with. The people who do most of the vaccine work with us are Bart Haynes at Duke University, and he does antibody work, Dan Baruch at Harvard, and he did the studies I showed you, and Tom Henke at Oxford. He does HIV work and now our Ebola work with us. And I can't say enough how much I appreciate this gentleman. So that's the end, and I'd be happy to chat with you guys.